Okay, hello everyone and welcome to BrainMap. This seminar series is co-sponsored by the P41 funded Center for Mesoscale Mapping housed in the Martinos Center. It's my immense pleasure to introduce our speaker today, Dr. Mikhail Shapiro. Dr. Shapiro is a professor of chemical engineering, an HHMI investigator, and director of the Center for Molecular and Cellular Medicine at Caltech. The Shapiro laboratory develops biomolecular technologies allowing cells to be imaged and controlled inside the body using sound waves and magnetic fields. These technologies enable the study of biological function in vivo and the development of cell-based diagnostic and therapeutic agents. Dr. Shapiro received his PhD in biological engineering from MIT and his bachelor degree in neuroscience from Brown. He conducted postdoc research at the University of Chicago and the University of California, Berkeley, where he was a Miller Fellow. Mikhail Awards, there are plenty of them, include the NIH Pioneer Award, the Packard Fellowship, the Pew Scholarship, the Willig Prize for Creative Promise, the Camille Dreyfus Teacher Scholar Award, and the Roger Zien Award for Excellence in Chemical Biology, and the list goes on and on. I would just like to remind the audience to please address any question you have using the Q&A box or raise your virtual hand. Dr. Shapiro, thank you so much for coming here today. The virtual stage is all yours.
All right, Or, thank you so much for inviting me. It's a pleasure to be here. I wish I could visit you guys in person, and someday I'll stop by Boston and say hello and see my many friends at MGH and Martinos. But yeah, thank you very much. And as we discussed before we started, I'm actually happy to take some questions during the talk. So if you have something, put it in the Q&A, and or we'll decide if it's appropriate to interrupt with the question, and we'll get going. So, all right. So in my lab, let's see if I can get this going. We develop technologies to image and control the function of cells deep inside the body. And this is motivated, first of all, by basic biology, because whether we're talking about neurons in the brain or microbes in the gut, or immune cells and cancer cells fighting each other somewhere else inside the body, to fully understand how these cells work, we need ways to study them within the in vivo context of all the anatomy and physiology and other cell types that are present there, and not just look at them under the microscope. And I know that at the Martinos Center, you guys completely buy into this vision. In addition, as we start to develop cells as therapeutic agents, and we all know about these beautiful successes like CAR T cells, there's interesting work with stem cells, with probiotics now that you can swallow that'll go into the gastrointestinal tract and detect inflammation or release anti-inflammatory compounds. I think as we start to put these cells into patients, it's going to be important to have ways to tell, where are they? Are they alive? Are they executing the functions that we programmed into them? And ideally also to tell them what to do based on their location. Now, communicating with cells in this way today is very challenging, because if you think about the best technologies we have available for connecting to cells, most of them are based on light. These are things like fluorescent proteins and optogenetics that derive a lot of their power from the fact that they're genetically encodable. So we have very good ways to plug them in intimately to what cells are doing. But the problem, as you know, is that light doesn't scale very well to intact organisms, even relatively small ones like mice, due to light scattering. And so it's hard to apply these techniques really within the in vivo context. And so we need to turn to alternatives. And we all know that there are forms of energy like sound waves and magnetic fields that can get in and out of tissues much more easily, giving us technologies like ultrasound and magnetic resonance that give us beautiful pictures like these ones of the developing fetus or the human brain. And the challenge is that while these technologies have really excel at anatomy and some aspects of physiology, they haven't been connected as well to the function of specific cells. And so that's the gap that we're trying to bridge. And the way we approach that in our lab is through biomolecular engineering, meaning that we find, we evolve, we engineer proteins and other genetically encodable materials that instead of interacting with light, can interact with sound waves and magnetic fields. And the work in our lab pretty much breaks down along the lines of sound waves and magnetic fields, and finding biomolecules and approaches that allow us to both image and control the function of cells inside the body. Now, I apologize to my friends who I see you're on here who are MRI experts, but today I'm going to talk completely about ultrasound. This is what our lab has been really focusing more on over the last few years, and I think it's probably less familiar to folks at the Martinos Center. So I wanted to purposely to highlight that. So I hope you'll keep an open mind. You know, I came to ultrasound after doing completely MRI early in my career. And when I started, you know, I thought ultrasound is just for imaging babies. It's not very interesting, but it turns out that it is. So I hope to influence you a little bit in that regard. So one of the reasons I love ultrasound is because it's both simple and powerful. It's simple in the sense that it's just based on sound waves that we transmit into a tissue. They propagate until they encounter an interface with something that has a different density or stiffness, like bone versus muscle. And then some of that sound gets reflected, and we can tell based on how long it was between transmission and reception, how far away, how deep this interface is. And then we can scan the beam left and right, and we can get an image like this. And you can see right away from this picture that ultrasound can go deep, right? This is tens of centimeters into the body. What may be harder to appreciate if you're not very familiar with ultrasound is that it's a very high performance technique. So routinely in our lab, without any special tricks and using off-the-shelf equipment, we get spatial resolution better than 100 microns, centimeters deep in tissue. It's very fast. Sound waves travel at 1,500 meters per second. So this round trip takes tens of microseconds. So we can get sub-millisecond temporal resolution. And there are two additional things recently that have made ultrasound even more appealing for biological research. One is the development of super resolution methods that are analogous to some of the ones used in optical super resolution based on stochastic signal sources that now allow you to get images like this one. This is a rat brain in cross-section in a live animal acquired with ultrasound, where the microvasculature within this brain is resolved at better than 10 microns spatial resolution. So really an impressive performance. And while being high performance, ultrasound is also quite accessible. So today, for $2,500, you can buy an ultrasound system where all of the electronics and signal processing is contained inside this handle, and you just hook it up to a smartphone and you're off and running doing ultrasound. So in principle, every biology lab in the world could be buying and using ultrasound to study the function of cells inside the body. So why aren't they? And I think the reason is that while these images give us, you know, nice access to anatomy and maybe some things like blood flow and so on, they're not really showing us the function of cells. So that's the gap that we need to bridge. And the inspiration for this comes from the history of fluorescent proteins, where there was an enormous leap in going from synthetic fluorophores to ones that can be genetically encoded, like GFP. And I'm sure you know about the revolution that fluorescent proteins have made in biology and enabled all kinds of new research. And so we want to do something similar for ultrasound. And what exists as synthetic contrast agents for ultrasound are these things called microbubbles. These are FDA approved. Because they have a low density, these are little bubbles filled with gas, because they have a lower density and are more compressible than blood and tissue, they will scatter sound waves inside the tissue. Okay, so if you inject them into the bloodstream, you can highlight the vasculature. And in fact, that was used to generate these beautiful images here. But the problem is, how are you going to connect these bubbles to the function of specific cells? I mean, these things are the size of a cell, and they're thermodynamically unstable. The gas wants to dissolve. And so they're really primarily used for blood imaging. And so the question that we've been kind of obsessed with for a number of years now is, can we find something in nature that has similar properties to these microbubbles that would produce ultrasound contrast and can be genetically encoded inside cells? And so luckily here, Mother Nature gave us a gift. It came from organisms like the ones pictured here. These are bodies of water that contain photosynthetic microbes that live in the water that had to solve the evolutionary problem of learning how to float, because they want to be near the top of the water to get enough sunlight for photosynthesis. Okay, so how does a cell float? I mean, normally cells sink, right? They're a little bit denser than water, but here, what they evolved to be able to become buoyant are these really beautiful intracellular self-assembled protein nanostructures called gas vesicles. Okay, intracellular self-assembled protein nanostructures. This is an electron micrograph of one of these that we've isolated from a cyanobacterium, and you can see it's this, you know, sausage-shaped structure that's a couple hundred nanometers in dimension. This is a protein, and what's special about this protein structure is that it's hollow on the inside and filled with gas. Okay, so you have a 2 nanometer thick shell that's enclosing this couple hundred nanometer compartment that excludes water, but allows gases from the surrounding media to partition in and out of this compartment. Okay? And when these cells want to float higher, they turn on the set of genes that encode the protein subunits that self-assemble into these nanostructures, make more of them, and so the cell becomes less dense and starts floating in the water. And when they want to sink, they can break these down with proteases and sink back down. So really a remarkable piece of evolution. And they were actually discovered back in 1895, and then people kind of forgot about them. But we came across them and we said, all right, you have a little protein that's filled with gas. Maybe we'll be able to see it with ultrasound. And in fact, that turned out to be true. So this is data from the first paper we published on this back in 2014, where all we did was take these proteins, isolate them, purify them from cyanobacteria, and embed them in hydrogels like agarose, which are acoustically transparent. So that's the black background that you see there. And then we embed these proteins at a series of concentrations. This is in the picomolar particle concentration range. And we saw this really nice ultrasound contrast. So that was exciting. And of course, one of the first things we said was, okay, can we see these inside of a living animal? And so we grabbed some mice and started injecting gas vesicles into the bloodstream of the mice. And what you expect for particles that are, in the size range, a couple hundred nanometers, is that they'll get taken up by the liver. That's the liver's job. One of its jobs is to clear these kind of particles from circulation. So we're doing ultrasound imaging. This is the liver up here. I'm going to start the video, and we're going to start injecting the gas vesicles intravenously. And over time, we see the accumulation of ultrasound contrast in the liver. And this was really exciting because to my knowledge, this was the first time that a biomolecule could be seen non-invasively with ultrasound inside of a living, breathing creature. And this really opened up a new field that we call biomolecular ultrasound that we've had a lot of fun over the last few years pursuing and just seeing where can it go? And, it starts with a lot of very basic questions about these structures, like, how do they interact with a sound wave, a pressure wave that's coming through? What happens to them mechanically? How do we play around with the genes that encode them? So one interesting thing, and also a complexity compared to a GFP, is that we're not dealing with one gene, but we're dealing with a group of genes that work together to form these gas vesicles. So how can we, first of all, transplant them into different organisms for heterologous expression, but how can we also play with them to change the properties, change the acoustics? And then how do we design ultrasound techniques that specifically interact with these gas vesicles to increase the sensitivity and specificity with which we can image them in tissue? And can we turn them into dynamic biosensors? So I'm going to show you a few examples of how we have pursued this. So one of the biggest questions and frankly, areas of skepticism when we started this work was whether we could ever express these gas vesicles in mammalian cells. And the reason this skepticism was warranted is because it's a pretty complex genetic program. So, previously people had taken, 2, maybe 3, maybe 4 gene cassettes and moved them from prokaryotes to eukaryotes, but they hadn't done a gene operon that's of this size. And the reason it's complicated is because the way bacteria and mammalian cells handle transcription and translation has significant differences. And it wasn't clear, whether all of these proteins, first of all, could fold and function as individuals within the mammalian environment. And moreover, whether they could find each other, be at the right stoichiometry, and assemble into these structures inside the cell. But luckily at Caltech, we have very courageous students who said, let's just do it and see if we can make it work. And it worked. And so here I'm going to summarize 4 years of work by a brilliant student and colleagues with this little black arrow right here, where we took all of these genes from the bacteria. First of all, showed that we can just co-transfect them together in the mammalian cells and get some gas vesicles to form. But then over time, figured out how to string these genes together using the latest tricks of synthetic biology into mammalian operons, where now you have these different genes that form gas vesicles that are strung together in a way that encodes them at the right stoichiometry so they can optimally assemble into gas vesicles. And we're using tricks from synthetic biology that a lot of people in the synbio community use, kind of these little elements that are borrowed from viruses or from bacteria or from other cell types, and we kind of make it work by putting together these kind of clusters. And if there are questions, I'm happy to go into it more. But the key is that now we have this genetic program that we can put into the genome of mammalian cells, like HEK cells here, and they start to form gas vesicles. So this is a thin section TEM image through a mammalian cell where you see these little bushels of gas vesicles forming inside the cell. And more importantly, this allows us now to watch gene expression in these cells with ultrasound. So we can connect the expression of what we call ARGs, or acoustic reporter genes, under the control of a promoter of our choice in these cells. And here, for proof of concept, we're connecting it to a promoter that's driven by a drug, doxycycline. And now we're taking living mammalian cells, embedding them in hydrogel, and watching gene expression in those living cells within this gel. And so as a function of how hard we drive this gene expression promoter with doxycycline, we see this increase in ultrasound contrast in these living cells. We can also follow the kinetics of expression. And so what does this look like in vivo? And for our initial test of this, we wanted to see if we could watch a pattern of gene expression within a tumor. And so these are subcutaneous tumor models in mice. And one of the things people know about them is that they're not homogeneously vascularized. You tend to have more vasculature towards the perimeter of the tumor rather than the core. And so in the perimeter, you expect to have a higher metabolism and therefore should have greater gene expression. And so we engineer these tumor cells to inducibly express the acoustic reporter genes. And we were hoping to see this pattern of gene expression within the tumor. And indeed we could. And so what we're looking at here is an ultrasound image where in grayscale is the anatomy. So you see the skin right here, the tumor is underneath the skin. And then in red scale is the ultrasound signal that's coming specifically from our gas vesicles through nonlinear imaging. And we see what looks like this ring-like pattern of expression with a hollow middle. And subsequently, we can section this tumor and look at it under an optical microscope, and we can see what that pattern of expression is. And over multiple animals, we see this nice correspondence in the pattern between what we see with ultrasound and what we see optically. Now, the difference is that the optical image is acquired after we euthanize the animal and cut it into little pieces to see what's here. Whereas on the left, with ultrasound, we're seeing live gene expression in a live animal, and we can continue to follow the dynamics of this, and we can resolve it here with 100-micron spatial resolution. We can also image bacteria. So there's a lot of interest in both fundamental studies of the microbiome, and one of the interesting things there is that people learn a lot about the microbiome by just looking at poop, which is great, but it's actually not very representative of the geography of what's happening. The species that you have in the small intestine versus large intestine versus the periphery versus the center of the colon and so on are very different, doing different things. So we need ways to image that in vivo. And then secondly, there's a lot of interest in developing bacteria as therapeutic agents, ones that are intended for use in the gastrointestinal tracts, ones that will home to the core of tumors. And I'll show a couple things about that later on. And so we also wanted to take these genes from the cyanobacteria that normally make them and see if we could move them into bacteria. And that also turned out to work. So this is a gene cassette we designed that we can express in E.coli, a common commensal microbe, where now you can see the cell is full of gas vesicles if we really push the gene expression. We can also put it under inducible promoters, and then we can watch gene expression now here in living bacterial cells. And as a proof of concept, we took some of these cells, we put them into the colon of mice, and we had a mixture of cells where some of them are expressing our acoustic reporter genes, and some of them, as a control, are expressing a luminescent gene. And I know lots of people use luminescence in biological imaging. And so now we have both of those types of cells within the colon, and they're in this spatial arrangement where the acoustic cells are towards the perimeter and the luminescent cells are in the middle. If we look at the ultrasound image, we see that expected spatial pattern inside the colon. If we look at the same mouse by luminescence, we see that there's signal coming from the gut, but the spatial information about the arrangement is lost because the photons are getting scattered on the way out. And this is an experiment that, frankly, reviewers asked us to do, but I'm glad we did it because it shows that if you use an actual non-photon spatially resolvable technology like ultrasound in this case, or the same would be true with MRI to image reporter genes, you can get this fine spatial resolution and not just see a blob of signal. One question you might have is how sensitively can we detect cells with this technique? And we've had over several years the evolution of different methods for doing this. And the most sensitive one is called burst. And the reason it's called burst is because it takes advantage of the destruction of gas vesicles. The way it works is that we send in an ultrasound signal, pulses at a low pressure. Then we suddenly step up the ultrasound amplitude that we're sending in so that our gas vesicles collapse, because if you apply enough pressure, it's, you know, it's a hollow shell structure like a ping pong ball. If you hit it hard enough, it's going to break. And when they collapse, they produce a really strong echo when that happens. But then immediately after that collapse, the gas that's inside of them escapes into the solution and dissolves. And after that, they're not producing any ultrasound signal, okay? And so we can take advantage of this unique temporal signature of an impulse of something turning on and turning back down to uniquely identify the gas vesicles versus all the other scatterers and interfaces within the tissue. And this allowed us to get to single cell detection. So here, these are individual E. coli or HEK cells that we're seeing with ultrasound. Now, you know, the pixels here are on the order of 100 microns. So we're not resolving individual cells, but we're detecting the presence of individual cells. So it shows us that this ultrasound technique can actually be quite sensitive. This is in vitro, and this is in vivo, where we're following the entry of salmonella into the liver, and we believe that these little signals here are coming from individual cells that have entered into the liver. So even with the things I just showed you, we're just at the very starting point. There are many, many things to do. We need to improve the reporter genes. You know, if you follow the analogy to GFP, nobody uses the original GFP anymore because it wasn't that bright, it wasn't that stable, et cetera. And so there are all these new and beautiful versions, and we'd like to do something similar with our acoustic reporter genes. We need to make the genes more convenient. For example, put them into viral vectors so they can be very widely used in a variety of cell types. We're also trying to create dynamic biosensors, improve pull sequences, and also think about burden and immunogenicity from cells that are making these things if we're trying to think about them for potential clinical applications. So I'm going to show you a little bit of these things here as well. So one of the approach we've taken to develop the next generation of our acoustic reporter genes is to revisit nature. I mean, Mother Nature gave us the gas vesicles in the first place, and we kind of selected ones from a particular species that was convenient for us to access. But we said, what if we go and look more systematically? And so what we could do is look at the evolutionary tree and sample different types of organisms to get a diverse sampling that all in the literature have been shown to produce gas vesicles. Take the genes from those organisms and see if maybe some of them work better in mammalian cells or E. coli, where we're trying to apply them. Now, you know, here we're looking at a larger number of samples that we're cloning and we're putting into bacteria and trying to image. So we needed a higher throughput assay to be able to look at this. And so what we did is build a robotic scanner. So here you have agar plates that have bacterial colonies on them, where each of these colonies is expressing a different genotype of acoustic reporter genes, of gas vesicles that we've acquired from a different organism, or in some cases we've mutated. And now we have this ultrasound that is being, a transducer that's being robotically scanned to go through and look at all of these colonies. And we can get ultrasound images from all of them. And we can look for ones that are brighter versus ones that are dimmer, and then follow up on that to see if, okay, maybe this one gives us a reporter gene cassette that produces nicer ultrasound contrast. And we can also test their response to different pressures, et cetera, et cetera. And so this has been really beneficial to us to speed up the engineering of our genetic system. So we looked at a bunch of different gene clusters. You don't need to look at the details of this. Cloned all of them into E. coli, did the screening, and we found some that seemed to be much brighter compared to what we had originally. These are our original genes right here, and now we have ones that the signal to background ratio is manyfold higher, and this has given us some significant improvements. So one of the things that we were looking for is to have an increase in nonlinear ultrasound contrast, which I'll explain more about in just a little bit. But that's how we can distinguish ourselves from background. And so our new generation of acoustic reporter genes 2.0 compared to 1.0 on a decibel scale here, have this great, 10 to 20 decibel increase in signal, right? So this is just from turning back to nature and saying, what else you got? And then taking advantage of that. Okay, so now we have these reporter genes that are brighter, and I just want to show you what kind of performance now we can get with them. So this is an example that I like, because one of the things people use ultrasound for, both clinically and preclinically, is to guide procedures. So people will do ultrasound guided biopsies where they're using it to image the tissue, and then they're going to stick a needle to do this. And so we asked whether we could do this in a genetically specific way. So if I have a tissue where I have some cells that are labeled based on their genetics to express acoustic reporter genes and some are not, can we go and access those cells and sample them, which we may want to do for experiments with multiple time points. So here we have a tumor. The tumor is chimera. So half of it is expressing the acoustic reporters and half is not. With normal B-mode ultrasound, you really can't see what's going on. With our nonlinear ultrasound, we can see exactly where it is. So this part of the tumor has the acoustic reporter genes, and this part does not. You can see this night and day kind of contrast. This is an overlay. And now in real time, we can just keep watching this with ultrasound while we bring a needle, and we can target it to either this part of the tumor, and it's kind of satisfying to watch the needle going in and grabbing some of the tumor for the biopsy. Or we could look at this side of the tumor, and then we can ask how well did we guide the biopsy by flowing the cells and flow cytometry, and we could see that we basically are perfect. That when we use ultrasound to guide our biopsies, we get exactly what we expect from the bright region versus the dark region. Another thing that we're interested now that we have this technology working pretty well is to look at interactions between tumors and brains for brain tumors, right? Meaning that while I have a tumor growing in the brain, how is it affecting the vasculature of the tumor, and how is it affecting the activity of the brain? And people have developed complementary techniques that I don't have time to explain, but I'm happy to talk more about in Q&A, where you can use ultrasound to map the vasculature of a tissue, and I already showed you some of that in the introduction. And then also people develop functional ultrasound to look at dynamic activity inside the brain, and that's something we also do in our lab. But so now we can add to those capabilities the ability to see acoustic reporters. So this is in a mouse brain with a tumor, and we can see exactly where the tumor is by ultrasound. We can follow the growth and evolution of that tumor over time, while at the same time looking at a map of the vasculature. How's the tumor affecting it? And then also look at neural activity. One of the other big advances that is making this technology more widely applicable is that we've figured out how to package those genes. We've made those gene clusters more compact, that we can put them into viral vectors. And so now we can label a wider variety of cell types. For example, here, T cells, macrophages, primary neurons. And so this opens up more ways that we can use this technology, both in research and potentially, now people are developing T cells as therapeutic agents, that this is something we may be able to apply in a therapeutic tracking scenario. We also improved the bacterial acoustic reporter genes. They're a lot brighter. So this is our new version. Compared to here, this is our old version, right? So, again, big night and day kind of differences. And so now I want to show you an example that I think is quite beautiful with tumor-homing bacteria. And, there's a number of groups in the world that are exploiting the fact that when you inject bacterial cells intravenously, normally they get cleared by the immune system pretty quickly, right? And these are not pathogenic bacteria. These are like friendly E.coli type of bacteria or attenuated salmonella. So normally they just get cleared by the immune system. But if you have a tumor that has an immunosuppressed core, these bacteria can infiltrate that core and establish a colony inside the core. And people are taking advantage of this to deliver anti-tumor therapeutics. And the great work on this from Sangeeta Bhatia's lab. I think this is from this illustration is from one of her publications. Tal Danino at Columbia is really doing a lot of groundbreaking work in it. But they can't see where these bacteria are after they inject them. Did they get into the tumor? Did they establish the colony? What is the spatial organization of the bacteria inside there? And so this is what we can see with ultrasound. So in green is the signal coming from our bacteria inside the tumor. We can see where they're distributed. In red is the vasculature using something called ultrasound localization microscopy, where we can get really high resolution images of the vasculature in the tumor. And the reason I like this image is because it really highlights the advantage of these bacteria, because normally when you administer drugs to a tumor, they have trouble getting beyond the vascularized regions, right? It's hard to get the drugs to penetrate into the core. Whereas the bacteria are exactly the opposite. They're wiped out wherever there's vasculature and they're accessible to the immune system. But in the core, they establish themselves. And now we can see this with ultrasound. And as I'll show you a little bit later, we can take advantage of this for therapy as well. Okay, so one of the other things that we've been very interested in is following in the footsteps of fluorescent proteins, where people engineered GFP and other fluorophores to sense dynamic signals. Okay, and so things like calcium or protease activity, or kinases and phosphatases, et cetera, where the intensity or wavelength of the emitted light changes in response to stuff that's dynamically happening inside the cell. And so can we do something like this for ultrasound? And what allowed us to do this is some fundamental work we did on the connection between the protein composition of the gas vesicles and their mechanics, and therefore their acoustics. And in particular, we found that there's this protein called gas vesicle protein C, or GVPC, that sits on the outside of the gas vesicle shell and acts as a mechanical stiffener. Okay, so when it's present, the gas vesicle shell is more stiff. When it's absent, it's more flexible. And the consequence of that is that when we apply a sound wave, which is a pressure wave that's coming through and pressurizing and depressurizing and pressurizing and depressurizing, is that the stiff gas vesicle won't deform very much, but the flexible one will buckle, just like an empty soda can if you squeeze on it. And the buckling is reversible because then you have compression and then you have rarefaction. So it's buckling back and forth. And this produces nonlinear signal that we can detect with ultrasound pulse sequences. And so this is an image where under nonlinear imaging, only this gas vesicle that's lacking GVPC and is more flexible produces this nonlinear contrast, whereas the normal linear ultrasound both look about the same. Okay, so what this lets us do is that now we can use GVPC as our hook for engineering gas vesicles to sense stuff. Okay, and the way we have done that initially with proteases is by engineering GVPC to contain recognition motifs that can be bound by proteases, and where the protease then can make a cut, and that takes our stiffening rod and breaks it in half. Or there are also proteases that are processive, meaning that they will recognize a sequence and then they'll chew the protein up like a spaghetti, and that'll also destroy the stiffening rod. And we hypothesize that by doing that, we can have the turning on of nonlinear ultrasound contrast because it makes the gas vesicle more flexible. And after a lot of engineering and optimization, where to put these recognition motifs, how to get them to be compatible with the structure, et cetera, we have sensors that work. So these are examples. This is in vitro, where we can sense different proteases. For example, the TEV protease, which is a viral protease, we can see when we add the protease, we're turning on the nonlinear signal. We have a sensor for human calpain. This is a calcium-dependent protease, where in the presence of calpain and calcium, you turn on this nonlinear signal, so we can sense that as well. And this is an example of one of these processive proteases that binds at the end and chews up the protein. This is ClipXP from E. coli, and you see this night and day turning on of ultrasound contrast. And this works also inside cells and in vivo. So we developed a system. Don't worry about all the details here. All you need to know is that we have cells where by giving this molecule arabinose, we're turning on this protease. Okay, so when we do that, we expect to see a turning on of ultrasound signal. And this is the entire system is now expressed inside the cell. So our sensor is genetically encoded. And indeed, we can see this turning on, that when we give the cells arabinose, we see this turning on of nonlinear signal. And we can see that even when the cells are inside an animal. For example, here they're inside the gastrointestinal tract. So this demonstrates that in principle, we can develop dynamic biosensors for ultrasound. This is for proteases. And of course, you can think about all the other things that we're now trying to sense. And I'd love to get ideas and feedback on other things in our discussion. I know that there are people who may work on kind of nanoparticle-based imaging at Martinos and other kind of contrast agents. So, gas vesicles don't have to be just genetically encoded. They can also be injected into the bloodstream, and they can be targeted to stuff. So we have convenient ways to functionalize their surface in a modular way. We've attached things that change their charge. We've attached things that bind to integrins. We've attached don't eat me signals that help them evade macrophage uptake. This is just an example of binding to a glioblastoma cell based on this kind of integrins. What happens to gas vesicles when they're injected into the bloodstream? Well, as I mentioned already, they get cleared by the liver pretty quickly. But it actually is interesting because they go into the liver, and there are lots of magnetic nanoparticles, for example, that get accumulated in the liver that tend to just stay there, right? So you can see the uptake job that the liver is doing, but of course, the other thing that the liver cells do is that they degrade biological particles, viruses, et cetera. But with inorganic materials, it's hard to see that because they're going to linger. But gas vesicles, because they're just made out of protein, we hypothesize that we'd actually be able to follow the lysosomal function of liver macrophages as well. And indeed, that's true. So when we simultaneously image gas vesicles in the blood and in the liver, we see that we see a clearance rate by which they get cleared from the blood. We see accumulation in orange here is a signal in the liver. We see an accumulation, and then over a slower time scale, there's a degradation. And we've done a lot of work in this paper to validate that what we're actually seeing is macrophage-based degradation. And we can follow now the kinetics of both the phagocytosis and the lysosomal degradation in vivo, and we've applied this to models of liver disease. Okay, I also want to mention, I know that there are a lot of people here who are interested in things other than acoustics, is that gas vesicles also have magnetic and optical properties. So everything I told you about the ultrasound is based on the difference in acoustic impedance between the air or gas and surrounding water. But gas vesicles are also different in terms of the magnetic susceptibility, right? You have air that's slightly paramagnetic versus water that's diamagnetic. And that actually produces, if you put that into a 7 Tesla MR scanner like we have, produces little magnetic field gradients around each of these particles that allow us to see them with T2, T2 star, and susceptibility weighted mapping by MRI. And one cool thing here is that we can take an MRI image and then we can just apply some ultrasound to collapse the gas vesicles, and that erases the MRI contrast. So now you have a before and after image that helps you tell these apart from microbleeding or other kind of things and get very nice clean images like this one here. And then they also have optical properties. And so the index of refraction is different. And that turns gas vesicles into very nice optical scatterers. And so we've done work where we've imaged them with OCT. And you can see that nicely in phantoms. And they actually, because they jiggle around, due to Brownian motion, you see this like dynamic speckle signal, which also enhances the sensitivity. You can inject them in the back of the eye, and you can get nice OCT contrast that also can be erased with ultrasound. So perhaps there are people in the audience who might be interested in these other applications. All right, so in last little bit, I want to tell you about, so I mentioned that we're interested in both imaging and control. Okay, and so far I've been talking about ultrasound-based imaging, but I want to say a little bit about control, because I think that's actually one of the other cool and powerful things about ultrasound is that everything I told you about so far has just been in one regime of what ultrasound can do. I call this the ping regime, where we're sending a brief pulse of sound, it's bouncing off of something, coming back to us and helping us form an image. But if we change our ultrasound transmission parameters, we can do all these other things. For example, if we have more extended ultrasound pulses and we have a gradient of them, meaning the pressure is inhomogeneous in our field of view, that can create a radiation force that will mechanically push objects in the medium. If we concentrate the energy at a focal spot and have these even longer waves, then we can deposit heat at those locations and heat things up. And this is used in the clinic for therapy. If we have some kind of bubbles present, and we use typically lower frequencies of ultrasound and higher intensities, we can cause bubbles to cavitate, to expand and contract, and that can be used for mechanical and therapeutic phenomena. And so we've been curious, to what extent can we couple each of these regimes, again, to the function of specific cells? And I just want to very briefly give you some examples of what we can do. So let's start with acoustic manipulation, this pushing things around. So it's been known for a very long time that there's something called acoustic radiation force. And one of the early examples of this was to send a sound wave into a capped tube. So the sound wave goes in, reflects off the end, comes back and interferes with itself to form a standing wave within the tube. If you have some particles that are within this tube, they will get pushed by that standing wave, depending on the particle properties, to either nodes or antinodes within the standing wave, and will concentrate. Okay, this is a paper describing it from 1866. You can do this easily at home. Just buy a little speaker from Amazon and a reflector, form a standing wave, and then take some styrofoam balls, and you can suspend them in midair. Okay? Fun things to do with your kids, perhaps. If you can afford it, you can also buy an array of speakers that you can get your kids to program so that they dynamically change the pattern of the sound waves within the field, and then you can get these particles to dance around in midair dynamically. So pretty cool. And of course, you want to know, can we do this with cells? And the thing that lets us do that is gas vesicles, because it turns out that the same properties that make them good at scattering sound for imaging are also allow them to experience a strong radiation force, meaning the difference in the density and compressibility of them relative to water. And in fact, we can calculate what's called the acoustic contrast factor, meaning how strong a force will they experience per unit field gradient, and compare them to other types of materials that people have tried to manipulate with ultrasound, like red blood cells, polystyrene, diamond, which is very, very stiff compared to medium. And what's interesting is that the magnitude of the force that the gas vesicles experience is expected to be an order of magnitude higher than the strongest of the other ones, because of the radical difference in both density and compressibility relative to medium, and because they're less dense and more compressible, whereas the materials on the left are more dense and less compressible, they'll move in opposite directions. So gas vesicles, and perhaps cells expressing gas vesicles, will go a different way from everything else in the water medium. And so we hypothesized that we could push gas vesicles around with ultrasound, and that we could take cells that express them and also push those cells around. I'm just going to show you examples with cells. So one of the things we can do is take some, in this case, bacterial cells, put them into a microfluidic channel. We've also fluorescently labeled them, so here we can see them with microscopy. And then within this channel, we're applying ultrasound to create a standing wave that's going to have a pressure minimum in the middle and pressure maxima at the sides. And we expect the gas vesicles to cause these cells to move towards the walls, towards the maxima. So watch this channel here. I'm going to turn on the ultrasound, and whoop, you see the cells moving. I don't know if that video played smoothly, but here, let me just play it one more time. Whoop, turn on the ultrasound, and the cells move to the edges. And this only happens when they're expressing our acoustic reporter genes. We can do more fun things, like have multiple nodes and antinodes within a channel, get the cells to arrange themselves, and then change the frequency so it changes the location of the attractors. And now the cells are dancing around, moving around within this medium. We can also concentrate ultrasound on a point, have a suspension of cells, get them attracted to that point. So you see this clump that's forming here after we turn on the ultrasound. And then we can take our transducer and we can translate it around physically and watch the cells get dragged around with it, right? So this is like an acoustic trap instead of an optical trap. And we're using that to manipulate, in this case, a clump of cells. And this potentially is also interesting for tissue engineering because there's a lot of interest in patterning cells within, say, a hydrogel as a starting point for an engineered tissue. And people have done all kinds of work to try to do that optically or with 3D printing type of methods that have some limitations. So we said, could we do this with ultrasound? So we collaborated with Pierre Fischer's group at Max Planck, where we take a hydrogel, we put cells in it, and before it has gelled, okay, so it's still liquid, we apply an acoustic hologram. This is something that Pierre specializes in, that can give us a particular shape. For example, the letter R in the acoustic field that's inside the gel. We let the cells arrange themselves in this shape while the gel is gelling. And after it's gelled, they're trapped. We turn off the ultrasound and the cells should be in that arrangement. So does that work? So this is for letter R pattern. And in fact, it does. So these are, as after we've done this, we see that we have our cells arranged in this letter R pattern. We can see optically. And as a bonus, because they're expressing gas vesicles, we can also see that pattern with ultrasound. Okay, the other thing I want to mention is cavitation. And here we're also going to take advantage of gas vesicles. What happens here is that I already told you that when we apply a high enough pressure acoustically, we can cause the gas vesicles to collapse. Okay. Now, normally when that happens, the gas that's inside of them just gets dissolved in the surrounding media within milliseconds and is gone forever. But if we're coming in with lower frequency and more extended pulses of ultrasound, then what happens is that the high pressure part of the pulse will cause this collapse to happen. Little nanobubbles get freed from the gas vesicles. But then the negative pressure part of the cycle will act as suction on these little bubbles and cause them to expand. Okay. And as they expand and contract over several cycles, they experience a force that causes them to coalesce with each other to form a larger bubble. And eventually, these bubbles get large enough that they can undergo inertial cavitation, which is a strong implosion that releases large mechanical effects and can be used for therapeutic purposes. Okay, sounds like a complicated thing, all these different steps, but it happens. And this entire thing happens on a microsecond timescale, which I'll show you in a second. Okay, so for the ultrasound experts, the way we prove this is by listening to what's happening within our sample. So we have this passive cavitation detector, we're applying focused ultrasound, and we can see these broadband signals that tell us that we're getting this cavitation to happen. And it specifically happens when we have gas vesicles present, not when we have other proteins present or when we just have buffer. But for the rest of us who are not deep ultrasound experts, we want to see this with our own eyes. And so what we did was we attached some of these gas vesicles to glioblastoma cells using those integrin binding domains that I told you about earlier. And we borrowed a 5 million frame per second camera from our aeronautics department. Okay, wait, sorry, before I show the video. 5 million frame per second camera from our aeronautics department, where we can look for the formation of these bubbles as we apply ultrasound and see what they do to cells. Okay, so here you see the texture of the cells. This video is slowed down by a millionfold. Okay, we turn on the ultrasound, so keep watching. We can't see the gas vesicles because they're too small, but over time, we start to see the formation and cavitation of bubbles within this medium. Okay, and so this is all happening on microsecond timescales, and so you have these rapidly oscillating bubbles that are undergoing cavitation, and what that does to these tumor cells is that it kills them. And we can see that by the entry of propydium iodide into the cells that happens after we apply ultrasound. So what we did was we converted our innocent little imaging agent into a bomb that we detonate on these cells. And Avino, who's a postdoc in my lab leading this work, said, okay, well, it would be even cooler if we can get the cells to detonate themselves, like make their own gas vesicles, and then we can get them to blow up. This was his artistic impression of that. And so what that would do is it could cause local damage within, say, a tumor, but also could release a payload from these cells that we can co-express, and then we lyse the cells. And indeed, we showed that we can produce broadband signals from cells that are expressing the gas vesicles. So meaning the cavitation works when the system is genetically encoded. We can release payloads. Here we're looking at bioluminescence that we've released out into the medium. And this works in both bacteria and mammalian cells. And so the way we applied this in a therapeutic context is with those tumor-homing bacteria. We inject them intravenously, we get them to home to the tumor. We can use ultrasound imaging, as I showed you earlier, to see where those cells are. So we say, all right, great. They have established a little colony inside the tumor. Now let's apply the focused ultrasound to cause this cavitation to happen, damage the tumor, and combine it with systemic administration of checkpoint inhibitors to awaken the immune system that in combination with this tumor damage would lead to a regression of the tumor. And we show that by doing that, we can dramatically reduce tumor growth compared to controls and extend the survival of the animals. Still lots to optimize, but I think it's pretty cool that we can kind of blow tumors up from the core using this. Okay, I'm going to mention one last thing very briefly, because I think it's time for Q&A, is that we can also use heat to activate things. And this is something that's used in the clinic, for example, to ablate uterine fibroids or to ablate the subthalamic nucleus inside the brain using focused ultrasound. And what we want to do is not ablate things, but to heat things more mildly and to use this small elevation of temperature as a control signal that tells cells to do something at their location. Okay, and so I'm going to, in the interest of time, I'm going to skip through a bunch of slides just to show you what we can do with this. So in the context of tumor-homing bacteria, we've shown that we can build gene circuits that respond to temperature that allow us to control the release of immunotherapy now locally inside the tumor. We're not administering it systemically, we're administering it locally. And with these tumor-homing bacteria, that now allows us to also reduce tumor growth, where now we're not blowing things up, but we're causing release locally. And the reason it's important to do this locally is because if you inject cellular agents, they often have off-tumor effects. Like they'll go to the liver, they'll go to the spleen, and they'll cause effects there. And so we want to overcome that. And then finally, that we can also do this with immune cells, where we showed that we can engineer T cells, for example, to respond to these small changes in temperature. And we can use that to activate tumor killing within those cells. I apologize, I'm running through this way too quickly for comprehension, but I hope you can check out these papers. Okay, so let me come to the conclusion. I thought I would put this provocative statement for all the MRI people in the audience and nuclear imaging people. Ultrasound is pretty awesome, and it is just becoming possible to interface ultrasound with cells using genetic tools. And this opens many possibilities and challenges. I want to thank and acknowledge the group of people that does this work. At the bottom of every slide, I acknowledge the people that did the particular projects. As you can imagine, this requires a really interdisciplinary team to do this kind of work. And finally, I want to put in a plug, if you're interested and curious about this kind of stuff, that we're hosting the first international symposium on biomolecular ultrasound and sonogenetics at Caltech in December. And we have an amazing lineup of speakers, and topics for this. I'm going to flip back to this in a second. And before that, also in Pasadena, Peter Caravan and I are organizing a symposium on the future of molecular MRI, and that's going to happen next month. And I hope you'll check that out as well. But since this is an ultrasound talk that I gave, I'm going to leave it here on this slide. So thank you again for having me, and I look forward to any questions.
Thank you so much, Professor Shapiro, for this amazing talk. And we now move to the Q&A phase, as you said. So I'd just like to remind the audience to please address your question using the Q&A, the chat box, or just raise your hand. I think we already have a question. Just a sec. Bruce.
Hey there. Maybe not surprising that I would have a question. I've got 1,000. Fantastic. Just so much progress and obviously huge amounts of hard work. So one question which kind of crosses the modalities, you showed this really nice example, ultrasound, MR, optical, kind of all with the same class of agents looking at the different biophysical effects. It's obviously not straightforward to, directly compare apples to apples in terms of sensitivity, but you must have some sense, kind of roughly, given, the underlying biophysics of each phenomena, what kind of different sensitivity regimes each of them are in, at least in the current state of affairs. That changes.
Yeah, that's a great question, Bruce. It looks like the ultrasound is the most sensitive. I think compared to, if we look at in the MRI case with the susceptibility, if we could just compare it to a super paramagnetic particle, we're not that close. I think it's interesting that just based on air, you can get that kind of contrast. And I think there is the advantage of being able to erase it with ultrasound, so you can subtract background. But, on a particle basis, with certain MPIOs, for example, you can get single particle, contrast with MRI. You are not going to get that with gas vesicles and MR. Whereas in ultrasound, because we can take advantage of dynamic changes in the contrast agent, we apply pressure, it collapses or it cavitates. That gives us an additional way to get more signal out of that. And optically, I think we haven't really pushed the sensitivity that much, so it's harder for me to say and make a compare, because actually there aren't that many contrast agents for OCT, let's say, right? So I think that still requires more work to figure out.
Yeah, I'm not surprised at all that the MR is not very good, and that you're going to beat them out. And I think what seems like a simple notion of just like turning it off, right? That just, it makes a huge difference in terms of your ability to deal with, kind of the 1/f noise and any kind of dynamic phenomena. It's the reason why we, with typical contrast agents, we look for 20-30% changes. In fMRI, 0.5% is fine, because we have this ability to turn things on and turn them off. And so that's a great advantage of the ultrasound techniques. I really get it. Really important.
Yeah, thanks, Bruce. And I would really welcome for people to explore the MRI applications further. The only reason we're not doing it is because the people joining my lab right now want to work on ultrasound. But
I don't blame them. If I was here, I'd be doing the ultrasound too.
Well, thanks. I do think, like, the turning off, I think they actually did make a big difference in our study. And one cool thing with gas vesicles from the point of view of turning off is that if you're imaging gene expression in cells, you can erase them and then the cells will make more. So the next day or 2 days later, you can do imaging again. So you don't completely lose the dynamics when you do it.
Super clever, super great. Really enjoyed it.
Thank you.
Thanks. Before we move to Shinyu, a quick question I saw in the chat just a sec, Shinyu. How small can gas vesicles be, and how small can they be detected by ultrasound from a chunky RAN?
Yeah, that's a great question. So we've made tiny ones that we call bicons that are on the order of 45 nanometers in size. That's probably the smallest that we have. Unfortunately, they don't produce as much ultrasound contrast because they don't deform as much. So we can't take advantage of these nonlinear imaging modes. But in the collapse mode where we break them, we do see sufficient contrast. And we're still trying to explore whether them being so small gives us advantageous properties in terms of biodistribution and so on.
Great, thanks. Shinyu, please.
Shin,
How are you?
hey, great to see you.
Yeah, so I have a quick question. Right? So I missed the first part of the talk, but you're showing this really interesting. You can collapse, these these gas vesicles, right? With ultrasound, I assume, right? So this is expressing cell, correct? Right. So in vivo, right?
Yes.
So I'm curious, have you tried any kind of pathology histology to showing that after collapse, right? How this potential damage is? And another question is, so I'm also curious, when you have the in vivo expression of these gas vesicles, where are their final, is the distribution of the vesicles is like always intracellularly, or there's any chance to extracellular, or and that could be just, I have a sense maybe you're working on that. That could be something very interesting in terms of dealing with lymphatic. Are you working on that or something?
That's very interesting. Yeah, that's very interesting. So so let me just answer the first question first. So when cells express the gas vesicles and we use collapse mode imaging where we collapse them, it's well tolerated by the cells. So in our papers, we've shown that the cells are fine in terms of viability and that they can re-express the gas vesicles afterwards. And that's because when we're doing collapse, normally we're using very brief pulses. So it's just applying the pressure to break the shell, but it's not doing the whole bubble cavitation that we do when we want to have a therapeutic output. And then in terms of extracellular expression, so far, all the gas vesicles we've seen are intracellular. And we haven't made an effort to try to get them exported in exosomes or anything like that. That I think could be interesting, but I think that would be a significant project to try to make that happen. We have injected them into the CSF in the brain and looked at drainage, and there is some interesting stuff that you see there that I'm happy to discuss with you offline.
Absolutely. Thanks.
Yeah, great to see you.
It's you too.
Great. So I think that for the sake of time, we're going to wrap up and thank our amazing speaker again and for this amazing talk, Dr. Shapiro. And goodbye, everyone, and next time in person.
Yeah, thank you so much.